I want you to imagine a scenario for a second. You go in for, like, a routine physical,
Uh-huh.
and your doctor runs a simple, relatively inexpensive blood test.
Right, just a standard lab draw.
Exactly. A few days later, the results come back, and the doctor sits you down to tell you that you have a severe terminal neurodegenerative disease. But here's the catch. You feel completely fine. Your memory is sharp.
Oh, wow.
Yeah. In fact, you won't actually feel or experience a single symptom of this disease for another 20 years. So the question is, do you truly have the disease yet?
It is a profound philosophical and medical question, honestly. And it is entirely upending how we think about human health.
Yeah.
Historically, a disease was defined by your suffering, by the symptoms you actually experience.
Right. You have a cough, you have cold.
Exactly.
Mm.
But medicine is rapidly shifting toward defining disease by the invisible, microscopic changes happening in your body decades before you ever feel sick.
Which is wild. You've shared a stack of intense medical research with us today, and we're taking a deep dive into it because it completely rewrites what we thought we knew about Alzheimer's disease. We're looking right under the hood at the pathology of dementia.
We really are.
Our mission today is to unpack exactly how microscopic proteins destroy human memory. We are going to explore the fierce debates rocking the neuroscience community, and we want to understand why the very definition of Alzheimer's is changing. Okay, let's unpack this. Because to understand why these proteins are causing such a stir right now, we actually have to travel back over a century to a German asylum.
We do. To really grasp the modern controversy, we have to start in November 1901. There's a German psychiatrist and neuropathologist named Dr. Alois Alzheimer, and he was working at the Frankfurt Asylum. He admitted a 51-year-old woman named Auguste Deter.
And reading through his clinical notes from the sources, her symptoms were just devastating. She was intensely disoriented. She developed this unfounded paranoia toward her husband.
Yeah, it was severe.
Her memory was failing rapidly, and her language skills basically disintegrated in a matter of months.
Dr. Alzheimer was absolutely captivated by her case. He kept incredibly detailed transcripts of his interviews with her. There is one specific exchange that really captures the tragedy of what was happening to her mind.
What was it?
He was observing her eat a meal of pork and cauliflower. He asked her what she was eating, and she replied, spinach.
Oh, wow. So she wasn't just struggling to find a word. Her brain was entirely misinterpreting the physical reality sitting on the plate in front of her.
Right. That is the terror of the disease. It doesn't just erase the past, it distorts the present. When Auguste died 5 years later in April 1906, Dr. Alzheimer obtained her brain for an autopsy.
He was working under Emil Kraepelin at the time, right? The famous psychiatrist.
He was. And Dr. Alzheimer decided to use a brand new tissue staining technique called Bielschowski silver staining on her brain tissue.
What does that actually do? Silver staining sounds like developing an old photograph or something.
It kind of is. It acts a bit like a molecular blacklight. By applying silver compounds to the brain tissue, certain hidden structures bind to the silver and turn black, making them suddenly visible under a microscope.
Oh, that's fascinating.
So when he looked at Auguste's tissue, he saw two distinct abnormalities that no one had ever systematically described together before.
The famous plaques and tangles.
Exactly. He saw dense extracellular deposits, what we now call plaques, sitting in the spaces between the brain cells. And then inside the neurons themselves, he saw strange, twisted, fibrillar structures, the tangles.
So he makes this monumental discovery. He is literally seeing the physical footprint of memory loss. He takes it to a psychiatric conference in Tübingen in November 1906. He stands up, presents these microscopic findings, and from what I read, the reaction from the audience was just total, absolute silence.
Total silence. They did not care at all.
Wait, really? Why?
The historical reports suggest the audience was just waiting for him to finish so they could hear the next speaker's topic. They thought he was describing some bizarre, one-off medical oddity.
That's unbelievable. I mean, it was his mentor, Emil Kraepelin, who eventually put the disease in his 1910 textbook and named it Alzheimer's disease. But he categorized it very specifically as a presenile dementia, and that prefix, pre, is where things kind of went off the rails.
Yeah, that single word birthed a 70-year medical mistake. Because Auguste Deter was only in her 50s, the medical establishment assumed this was a rare disease that only struck middle-aged people.
So they completely separated it from older people getting dementia.
Exactly. They explicitly separated it from senile dementia, which was their catch-all term for the memory loss commonly seen in older adults.
This is where we have to push back on this deeply ingrained cultural idea of normal aging. For 70 years, doctors looked at an 80-year-old losing their memory and said, well, that's just normal aging. It's senile dementia, brain wear and tear.
Right, they just accepted it.
They thought Alzheimer's was just aging sped up and happening too early. So how did we finally realize they were the exact same thing?
It wasn't until the late 1960s and 1970s that pathologists started routinely applying those silver stains to the brains of 80-year-olds who died with so-called senile dementia. What they found shocked the field.
Let me guess. The exact same plaques and tangles.
The exact same amyloid plaques and tau tangles that Dr. Alzheimer found in Auguste Deter.
Meaning they weren't two different conditions at all. Alzheimer's disease wasn't a rare medical curiosity. It was a leading cause of death in the elderly that we had just been ignoring as normal.
Exactly. Overnight, it transformed into a massive public health crisis, which forced the scientific community to ask a much harder question. How do these two microscopic structures actually dismantle a human mind?
Well, let's break that down, starting with the molecules. From the sources, it seems like the plaques form from a kind of biological garbage processing error. There is a protein called amyloid precursor protein, or APP, that sits in the membrane of the neurons.
Yeah, so the brain is constantly building and repairing the physical connections between neurons. APP seems to play a role in that maintenance, but proteins don't last forever.
Right, they degrade over time.
Think of APP as a long string that regularly needs to be cut up, cleared out, and recycled. To do this, the brain uses enzymes called secretases, which act like tiny molecular scissors.
So if the normal scissors, an enzyme called alpha secretase, cuts the APP string, the resulting fragments just dissolve and get cleared away, no problem.
But in Alzheimer's, a different set of scissors takes over. First, beta secretase makes a cut, and then gamma secretase makes another cut. They slice that APP into a very specific, slightly too long peptide fragment called amyloid-beta.
Specifically, a 42 amino acid version known as a beta 42, right?
You got it.
And the defining feature of a beta 42 is that it is incredibly sticky.
It is entirely hydrophobic, meaning it actively repels water. So instead of floating away to be cleared in the brain's fluid, these sticky fragments start clinging to each other.
Like magnets.
Exactly. First, they form small, soluble clusters called oligomers. Then they form longer chains, and finally they fold into those dense, hard, insoluble amyloid beta plaques that Dr. Alzheimer saw sitting outside the brain cells.
And the current thinking isn't necessarily that the hard plaques are the biggest problem, right? It's those smaller, soluble oligomers floating around beforehand.
Yeah. Researchers increasingly believe the oligomers are the most toxic element. They drift through the brain and physically interfere with the synapses, the tiny gaps where neurons communicate.
Oh, I see.
If you're trying to learn a new name or remember where you parked, your neurons need to fire and strengthen that synaptic connection. The amyloid oligomers essentially gum up the connection, preventing the memory from forming in the first place.
Okay, so that's the extracellular problem, the plaques outside the cell. Let's look inside the cell at the tau tangles.
Inside every healthy neuron, you have a protein called tau. A neuron isn't just a round sphere. It has a very long, delicate arm called an axon that reaches out to connect with other cells.
Like a wire.
Right. And to transport nutrients and chemical messengers down that long arm, the cell uses a microscopic railway system made of structures called microtubules.
And tau's job is to act like the wooden railroad ties that hold those tracks together.
That is its normal function. But in Alzheimer's, that tau protein gets what we call hyperphosphorylated.
Whoa, hyperphosphorylated. Hold on. For those of us who haven't taken high school biology in a while, what is actually happening to the protein there?
Fair question. A phosphate group is just a small chemical tag with a strong negative charge. When enzymes incorrectly attach way too many of these negatively charged tags onto the tau protein, it completely changes its shape.
Oh, so it physically distorts.
Yes. Think of it like trying to force two magnets together with the same polarity. The repulsion forces the tau protein to physically detach from the microtubule tracks.
So the railroad ties just pop right off.
They pop off and they start clumping into twisted pairs. Those are the neurofibrillary tangles.
Right.
And because the tau has fallen off, the microtubule tracks completely disintegrate.
If the tracks disintegrate, the cell can't move supplies anymore.
Exactly. The neuron essentially starves to death at its far end. Eventually, the entire cell dies. But the tangle itself is so dense and insoluble that the brain can't easily break it down.
So it just stays there.
It just gets left behind in the tissue, sitting there like a microscopic protein tombstone long after the neuron that created it is gone.
I want to try an analogy here to make this really concrete. The amyloid plaques outside the cells feel like a massive garbage strike in a city. The toxic trash bags are piling up in the streets, blocking the road, stopping communication between the houses.
That's a great way to picture it.
But the tau tangles, that is a disaster happening inside the house. The tau is like the internal load-bearing walls of your home. When the tau fails, those walls slowly rot away until the entire roof just caves in on itself.
That captures the dual nature of the pathology beautifully. What's fascinating here is that this internal collapse doesn't just happen randomly all over the brain at once. In 1991, two neuropathologists, Heiko and Eva Brake, mapped this out.
Oh, right.
They proved that these tau tangles follow a relentlessly predictable staged route of destruction.
The break stages.
Yes. The tangles almost universally originate in a very specific area deep in the brain, the entorhinal cortex and the hippocampus.
So you're saying the disease attacks the brain's main memory indexing hub first. Is that why short-term memory is always the signature first symptom?
It is a direct physical consequence of where the tangles begin. People often think Alzheimer's memory loss is just forgetting where you put your keys, like a retrieval issue.
Right, like the file is there, but you just can't find it.
Exactly. But because the hippocampus is under attack, it is actually an encoding failure. The information never gets written down onto the brain's hard drive in the first place.
Which perfectly explains Augusta Turr. The visual information of "I'm eating pork" couldn't be indexed, so her brain just grasped at spinach.
Precisely. And then as the break stages progress, those tangles spread outward into the neocortex, the wrinkly outer layer of the brain, which is when you see the profound personality changes, the language breakdown, the loss of spatial navigation.
So it marches across the brain over time.
Yeah. If a pathologist looks at a brain at break stage 1 or 2, the person probably had no noticeable symptoms in life. By stage 5 or 6, the neocortex is heavily invaded, correlating tightly with full-blown severe dementia.
Wait, if the tangles and the plaques are doing all this destruction, then whoever has the most plaques in their brain must automatically have the worst memory, right? If they cause the disease, more plaques should equal a worse condition.
That was the logical assumption governing the field for a long time. But the science showed us something entirely counterintuitive. In the 1990s, a researcher named Robert Terry led a study looking at this exact correlation.
And what did he find?
He found that the raw count of amyloid plaques in your brain is actually a terrible predictor of how cognitively impaired you are.
What was the predictor then?
The single best predictor of cognitive decline was the degree of synapse loss, the physical vanishing of the connections between the neurons. By the time a patient has moderate to severe Alzheimer's, they have lost roughly 25 to 35% of their synapses in the cortex and hippocampus.
That's a massive amount of the network just gone.
The plaques and tangles are present, but it's the severing of the network that actually drives the symptoms you experience.
And this brings us to one of the most incredible studies in the sources we reviewed, the 1986 NUN study by epidemiologist David Snowden.
It remains a landmark piece of longitudinal research. Snowden followed a convent of the School Sisters of Notre Dame. Because they lived in a highly controlled environment, he had access to their incredibly detailed life records, their early writings, and crucially, they all consented to donate their brains for autopsy upon their passing.
What they found at autopsy was stunning. They found some sisters who had massive plaque and tangle burdens in their brains, like pathology that looked exactly like end-stage Alzheimer's.
Yeah, brains full of pathology.
But while they were alive, these specific women showed absolutely zero measurable cognitive impairment. They were teaching, debating, and writing brilliantly right up until the day they died. How is that even possible if the load-bearing walls are collapsing?
It gave rise to a crucial concept called cognitive reserve. It suggests that things like high levels of education, lifelong mental engagement, and excellent vascular health act as a physical buffer.
So they basically outbuilt the disease.
Pretty much. These women had built up such dense, robust neural networks over their lifetimes that their brains could essentially route around the damage. They could tolerate a massive pathological burden without ever showing the clinical symptoms.
Wait, if you can have a brain completely full of amyloid plaques and still be sharp as a tack, doesn't that completely destroy the idea that amyloid plaques cause Alzheimer's in the first place? Is amyloid actually the trigger, or is it just a bystander that shows up at the scene of the crime?
You have just stumbled into the biggest battleground in neuroscience, the amyloid cascade hypothesis. For 30 years, this theory dictated almost all research funding.
Oh, wow.
The idea is that amyloid accumulation is the initiating spark that triggers a cascade of everything else. The tau tangling, the immune system inflammation, the synapse loss, and finally, cell death.
To be fair to the theory, The sources point out some massive genetic evidence supporting it, especially with early onset familial Alzheimer's disease, which tragically strikes people in their 30s and 40s.
Those cases are rare, but they are genetically deterministic. They are driven by inherited mutations in 3 specific genes, the APP gene itself, and 2 genes called PSEN1 and PSEN2.
I remember reading about APP, but what do the PSAM genes do?
They are actually the core components of that gamma secretase enzyme we talked about earlier, the rogue molecular scissors that make the final cut to create the sticky amyloid.
Ah, okay.
Every single one of those early onset genetic mutations does the exact same thing. It ramps up the production of that toxic A-beta 42 fragment. That is incredibly powerful proof that overproducing amyloid is sufficient to cause the disease.
There's also a very profound connection to Down syndrome that acts as a sort of tragic natural experiment for the amyloid theory.
The gene that produces amyloid precursor protein sits on chromosome 21. People with Down syndrome have trisomy 21, meaning they carry an extra copy of that entire chromosome.
So they literally have more of the APP gene.
Because they have a third copy of the APP gene, their bodies produce elevated levels of amyloid their entire lives.
Creating a direct dose-response effect.
Sadly, yes. Due to that extra gene, virtually all people with Down syndrome develop amyloid plaques by their late 30s, and a majority will go on to develop clinical Alzheimer's dementia decades earlier than the general population.
But for all that genetic proof, the amyloid hypothesis has taken some massive hits in the real world, particularly when pharmaceutical companies try to turn the theory into a cure.
This raises an important question. What happens when a scientific theory works perfectly in an animal model but fails in a human clinic? For years, drug companies developed antibodies designed to hunt down and clear amyloid out of the brain. A famous example is a drug called solanezumab, heavily tested through the 2010s.
And according to the data, the drug did exactly what it was engineered to do. It successfully cleared the amyloid from the patient's brains.
It did.
But in massive phase 3 clinical trials, the patient's memories didn't get better. Their cognitive decline wasn't meaningfully slowed at all.
That massive disconnect, the fact that we removed the plaques but the patient continued to decline, deeply fractured the field. It fueled a resurgence of researchers looking elsewhere, suggesting that perhaps tau or brain inflammation or metabolic dysfunction were the true drivers.
It also didn't help that the amyloid field suffered a massive, highly publicized controversy. We have to talk about the 2006 A-beta 56 scandal. What made this specific paper so important?
In 2006, neuroscientist Sylvain Lesné published a high-profile paper in the journal Nature. He claimed to have identified a very specific subtype of amyloid-beta oligomer, which he named a beta 56.
Okay.
The paper claimed this specific molecule caused memory impairment in rats entirely on its own, independent of the larger plaques. It seemed like the smoking gun the amyloid hypothesis needed.
It became one of the most cited studies in Alzheimer's research. It guided millions of dollars in NIH funding and heavily dictated the direction of pharmaceutical research for over a decade.
But in 2022, a neuroscientist named Matthew Schrag published a forensic image analysis in the journal Science. He identified what appeared to be extensive, deliberate image manipulation across dozens of Lesné's papers, prominently including the foundational 2006 Nature study.
Wait, this is crazy. Are you saying an entire decade of research direction and millions in funding might have been influenced by photoshopped images? Now, to be clear to you listening, the sources report on this as a massive research integrity investigation, and we aren't weighing in on the motives or taking sides here.
Right, absolutely.
But critics of the amyloid hypothesis point to this and say it diverted critical funding away from other promising theories for years.
It is undoubtedly one of the most consequential integrity cases in modern neuroscience. Now, it is crucial to remember that the broader amyloid hypothesis doesn't rest solely on that one paper. The genetics of early-onset Alzheimer's and Down syndrome remain factual.
Right, the foundation is still there.
But the scandal certainly added intense fuel to the fire for critics of the amyloid-first approach.
So, what does this all mean for the people actually living with this today? I want to pivot from the laboratory drama to the real world, where there are an estimated 55 million people worldwide navigating dementia right now.
To do that, we first need to clarify some terminology that causes immense confusion in clinics every single day. Dementia is an umbrella term. It is a clinical syndrome describing a progressive cognitive decline severe enough to impair a person's daily life.
Got it.
Alzheimer's disease is simply the most common underlying cause under that umbrella, responsible for about 60 to 70% of all cases.
So it's like saying someone has a fever versus saying they have the flu. The The fever is the symptom you experience. The flu virus is the underlying physical disease causing it.
That is a perfect way to look at it. There are other diseases under the dementia umbrella. There's vascular dementia, caused by cumulative mini-strokes or poor blood flow to the brain.
Okay, that makes sense.
There's also Lewy body dementia, which involves a totally different misfolded protein called alpha-synuclein and often presents with visual hallucinations.
You also hear a lot about the APOE gene, specifically the APOE epsilon 4 variant. If you get a genetic test and find out you have this gene, is it a guarantee you'll get Alzheimer's?
It is not a guarantee.
Huh.
APOE epsilon 4 is the strongest common genetic risk factor for the late-onset sporadic form of the disease. If you inherit one copy from a parent, your risk roughly triples.
Wow, triples.
Yeah, and two copies raise the risk significantly higher. But unlike the early-onset genes we discussed, having APOE epsilon 4 just heavily shifts your probability. Many people with the gene never develop the disease, and many without it do.
Which brings us to the treatments. For decades, doctors have relied on drugs like Donepezil. But from what I understand, those don't actually fix the underlying disease. They just temporarily mask the symptoms by boosting certain chemical messengers in the brain.
Exactly.
But recently, we have seen the controversial arrival of disease-modifying anti-amyloid antibodies, drugs like aducanumab, lecanemab, and donanemab. Do these actually work?
The clinical benefit is incredibly modest and highly debated. Aducanumab was approved by the FDA in 2021, but the agency's own advisory committee voted overwhelmingly against it because the efficacy data was contradictory. 3 panel members actually resigned in protest over the approval.
The newer ones, lecanemab and donanemab, did show a statistically significant slowing of cognitive decline in trials, though. But you have to weigh that small benefit against some very real risks.
These drugs can cause a side effect known as ARIA, which stands for amyloid-related imaging abnormalities. Put simply, it means brain swelling and microhemorrhages, brain bleeding.
Why would clearing amyloid cause the brain to bleed?
Because the amyloid protein doesn't just deposit in the brain tissue between the neurons. It also builds up in the walls of the brain's blood vessels, a condition called cerebral amyloid angiopathy.
Oh, I see.
When these powerful antibody drugs attack the amyloid in the blood vessels, the vascular walls can weaken and become leaky.
So patients and their families are left in this grueling position, trying to weigh a potentially small delay in memory loss against the risk of literal brain bleeding. It is a heavy burden, especially when you realize the vast majority of Alzheimer's care falls on unpaid family members, mostly daughters and spouses, who suffer enormous personal, physical, and financial tolls.
It is a devastating reality. But while we wait for safer, more effective drugs, the sources heavily emphasize that lifestyle interventions are proven to help right now. Controlling your blood pressure, managing diabetes, exercising, treating hearing loss, and staying socially and mentally engaged can significantly reduce your risk of developing clinical dementia.
So you can actively build up that cognitive reserve we saw in the NUN study.
Yes, exactly. If we connect this to the bigger picture, all of this science, the plaques, the tangles, the genetics, culminated in a massive shift in 2018. The National Institute on Aging and the Alzheimer's Association published a research framework that formally pushed to redefine Alzheimer's disease strictly as a biological entity.
Meaning, the medical field wants to define you by your biomarkers, not by your memory.
Under this new framework, if a doctor finds amyloid and tau biomarkers in your spinal fluid or on a specialized PT scan, you technically have Alzheimer's disease, even if you are cognitively completely normal and have 0 symptoms. They consider it a preclinical stage of the disease.
Which brings us right back to the scenario we started with. With the rapid rise of cheap, highly accurate blood-based biomarker tests, this biological redefinition is leaving the research lab and entering your primary care doctor's office. And it leaves you with a deeply personal question to mull over. If you could take a simple blood test tomorrow that tells you whether you have microscopic amyloid plaques, technically handing you an Alzheimer's diagnosis 20 years before you ever forget your keys or fail to recognize a loved one, would you actually want to know? And if you did know, how would that invisible biology change the way you live every single day you have left?